many of you know, the mission of my podcast is to host founders of the most disruptive companies in the world and to explain what they do to my audience in a format that's digestible. Beam Therapeutics is pioneering the use of CRISPR-based editing to develop a broad portfolio of advanced genetic medicines. Your groundbreaking base editing technology allows us to make permanent, specific edits to single bases in DNA and RNA without cutting the strands of the DNA helix. So today we'll hit on some of the revolutionary science going on at Beam Therapeutics. We'll chat with John Evans, the CEO of Beam Therapeutics, about his journey prior to his arrival at Beam and what life has been like since joining the Beam Therapeutics family. So I have so many questions I would love to ask, but for the sake of time tonight, I boiled it down to some of the most burning questions. So it's my honor to have you to join Making the Cut podcast, powered by Pounding the Table Network. And congratulations to Beam on your recent INDs as you make the next step to bringing potential curative therapeutics into the clinic. We've all been waiting patiently for that announcement to come, John.
Thank you. It's great to be here, and I really appreciate this chance to talk with your audience, and appreciate everything you're doing to just try to bring more education and awareness to the space. It's really exciting.
Awesome. So John, without further ado, the first question is that I believe it was around 2017 where you met with David Liu at his office at Harvard. And base editing really captured your imagination. So at the time, you were doing some work for Arch Venture Partners. You mentioned word for word that it was clear right away that Beam was a potential rocket ship. Dr. Liu is also on record saying that at the moment he met you, he knew you were the right guy for the CEO position. So are you able to share with us some of those preliminary conversations that took place with Dr. Liu? What do you think he was looking for in a leader? And what was it that really attracted you most to Beam's base editing platform from the get-go?
Yeah, great. So I love a good origin story. So maybe I'll go way back just to start. So I was actually originally an English major in college. So I share that only to give your audience a sense that it isn't just science PhDs that can navigate this space. But it's been quite a strange trip for me. But I learned early that I love medicines, and I really wanted to work closer in therapeutics. And then also, as I got into the pharma industry, I started to realize that I wanted to work in smaller and smaller companies, because that was just really where you could see it all come together, and the moving faster and the innovation level was going up, right? So the most recent company before Beam is probably worth a comment, because it gives you a good sense of why Beam resonated so much with me. So that was Agios. So Agios is a small molecule cancer company that targeted metabolism. It's looking after cellular enzymes and metabolism, and trying to treat cancer with that. And amazing company. And we ended up focusing on genetic mutations in some of these enzymes. And we had drugs that literally would turn off those mutations. So you're basically fixing what's wrong. Right. And we call that precision medicine, because what that means is you basically know who to treat when you go to the clinic. So you're not spending phase 1, phase 2, phase 3 searching for signal and then trying to statistically show that your drug has some activity. You're right away treating patients who have the mutation, and your drug should fix it. And so sure enough, very early in phase 1, we saw activity. We saw complete responses in patients with acute leukemia who should be dead in a couple of months otherwise, right? And what that means is that you now really know you have something. And so now you can work with the FDA and with patient groups to move as fast as possible. And so we ended up getting 2 drugs approved with this sort of similar mechanism in less than 4 years from IND. Now that's like light speed, but I think in general, these precision medicine approaches are always going to be faster than the 8 to 10 years that it normally takes. And you just have higher confidence in what you're doing biologically. So for me, that was really powerful. The frustration at Agios was that in small molecule world, if you do it once, it's great, lightning strikes, but then to do it again, you're basically starting from scratch. New targets, new chemical scaffolds, and it's just the whole journey and all the same risks. So that was frustrating. Agios is going to get there. They're going to get some new drugs soon. But so I left Agios. It was the right time. And so I got involved in Beam. And so, as you said, I met David Liu, and I'd never met him before. And it was an incredible experience. And he talked me through base editing. And I did not know a lot about CRISPR at the time. And I certainly didn't know about the nuances of Gen 1 nucleases and scissors versus what base editors can do. And I know we'll talk about all of that. But what really struck me was 2 things. One was that this was sort of the ultimate precision medicine, right? Because now every patient we could treat with this system, we will know who they are based on their genetics. And then we'll have a drug that can potentially reverse those genetics back to normal. So just like my Atlas experience, these were going to be clinical paths where we really know who to treat, and we really think it should work, and it should work early, and it should set up a fast path to getting an approval and getting it to patients. But even better, it's a platform.
Right.
Because these are basically programmable. And as I know your audience knows, the beautiful thing about CRISPR and the reason why it was such a breakthrough over zinc fingers and TALs and other things that come before it is that easy programmability. The guide RNA encodes the address in the genome where we want to search and then bind, change that little element, and you have an entirely new medicine. And yet everything else about the medicine could be exactly the same: the manufacturing, the regulatory package, all of that. And so that meant that with base editing, if we could do it even once, we could do it 10 or 20 times relatively quickly. And so that really was what captured my imagination. So I couldn't sleep that night. I was sort of just thinking constantly about what was possible here. And I think the elegance too of being able to think about single letter changes within the code of DNA and using that to basically reprogram the outcome of what the gene is coding for, whether you're fixing it or just altering it or modifying it functionally. The possibilities are really endless. And that's certainly where I think David and I did hit it off very early. I think sharing a common love of science and a common sense of urgency, I think, to get these technologies to patients as quickly as we can, and a little bit of shared just geeking out on what was possible with the technology, and we went off to the races. And of course, we were joined by Feng Zhang from MIT in the Broad, and then Keith Jung from MGH, just incredible scientists in the CRISPR field, and that was sort of the original group that helped set up the company.
And so, John, bringing novel medicines into the clinic comes with a great deal of responsibility. While Beam does away with the double-strand break, it's certainly not free of other precision chemistry risk factors. So, bystander editing is editing of one or more non-target bases in addition to the target base in the activity window of a base editor. And translocations are another risk factor, which leads to chromosomal abnormality due to the double-strand breaks. So, at Beam, how do you think about these risk factors? What have you seen from preliminary animal models, assay tests, and how far do you think we've come in our abilities to study cell changes at the single base resolution?
Yeah, great question. So maybe to answer this question, let me do now the mini PhD for your audience in terms of what base editing is. And I know a lot of people already know this, but it's probably worth doing.
Right.
So, when I think about all these editing tools, I think of them actually in the context of the general push for gene therapy, which is we want to have one-time curative treatments. And with gene therapy like AAVs or lentivirus, you're basically adding some extra gene into the cell. You can't control where it goes, but it'll just go somewhere, but hopefully it expresses, and that may help. The idea has always been it would be better to do gene editing. Why don't we just work with the genes that are already there and hopefully fix them or correct them? And so the first generation of gene editing tools had one really important breakthrough. And this was, again, zinc fingers and then talons and then famously CRISPR. And that was how you target. So if you think about it, every one of your cells, A, G, C, and T, has 3 billion letters in it, and you have to find a single address out of those 3 billion. I mean, that is an incredible challenge. And those different tools can do that. And it's really quite remarkable how they do it. Obviously, CRISPR, the ease of it and the flexibility is why it's so popular, but all of them have that ability. So that's amazing. The drawback in Gen 1 is once you get there, the only thing you can do is cut. Okay, so you make a double-stranded break, which is like a cleavage. And then really at that point, your job is done and you're leaving up to the cell what the cell will do next. And the cell basically doesn't like that. It's a genotoxic event. So it'll put the pieces back together again as quickly as it can, but it does so with damage. So you get random insertions or deletions of letters at that target site, basically scrambling the sequence at random. And so that will basically disrupt a gene, and it's good for knocking things out. But obviously the randomness of that and the lack of control isn't so easy to control the outcome of the edits. You don't have that precision control. And so that was really Gen 1. And obviously that's moved forward, and now there are places where you can knock something out and have some therapeutic benefit, and those are the programs that we see now moving forward. Beam really comes out of Gen 2. And so it was some scientists in David Liu's lab, including Alexis Comer, Nicole Gaudelli, who said, isn't there a better way to do this? And so what we came up with was base editing. And so what base editing does is it uses CRISPR for that same targeting ability we love so much. But once we get to the target site, we open the DNA up and we use a deaminase, which is a naturally occurring chemical enzyme, to edit one letter to another. Okay, so we make this very precise single letter change we don't need to make the double-stranded break. So that's the critical answer. So now in thinking about your question about sort of what are the consequences of our editing, what are the kinds of off targets we think about. So the first important breakthrough is that we don't make the double-stranded break, and that's really critical. And so double-stranded breaks have lots of consequences in cells, and the more of them you make, the worse it gets. So there's damage pathways that turn on, the cell viability can get worse, and particularly you can end up with chromosomal changes, because if you think about it, the cell doesn't always know which pieces go where. And so sometimes it can make that mistake. And so if you make a mistake, you'll get complex, novel chromosomal aberrations, and that can be quite concerning. So generally, I am of the opinion that moving away from double-stranded break editing whenever possible is going to be preferable. And so that's one thing that base editing does. Now, you noted one of the features of the way base editing works is there is something called bystander editing occasionally. And so the way that works is basically what base editing is doing is it's actually opening up a little bubble in the DNA, and it's a short window of about 4 to 5 bases wide. And the deaminase we use will recognize only one kind of base. So if we use an adenosine deaminase, it will only recognize an A, cytidine deaminase will only recognize a C. And so if there are more than one A or C in that window, you can potentially edit multiple bases. And there's no particular way to stop that other than to shift the window or re-engineer your editor, which we actually can do. So our job is to target the base that we want to target in the middle of that window, and then see if there's any bystander edit that we have to be concerned about. The bottom line is bystander editing really has not slowed down any of our programs and is not something that we're particularly concerned about. And if you think about it in a couple of ways, I mean, if we're trying to sort of repair a point mutation that's causing a disease, let's say, so you have a broken gene and you want to fix something that's wrong, okay? So let's say we make that edit successfully. If we edit something else, then you have to see, does it have any consequence? And actually, every third letter we edit has no consequence because of the way that the amino acid code is constructed. And so those are just synonymous changes. And then if there are other edits, they may be at very low levels, and most of them are inconsequential to the function of the gene. You've still restored the function of the gene. In the worst case scenario, if it was going to disrupt the function of that gene, you've just done a round trip. You fixed it and then you broke it again. so there's not going to be like a toxicity question or a safety question. You just failed to cure that cell. And so, you know, these are things that you can basically test very quickly in vitro and identify whether that's something to be concerned about. And if so, you can engineer it and work on it. The thing to stress is to compare that situation to the nuclease again, right? So we have a very well-defined and precise editing profile, which is generally either just the base we care about, or if we have one of these bystanders, we have to think about the bystander. But we're still getting the same pure product every time. The nuclease, every single cell is getting a different outcome. You're getting tens or hundreds of different unique alleles based on the random outcome of that indel process. And so we kind of report indels in aggregate. We say we got 80% indels, but the reality is indels is code for a wide rainbow of different alleles, right? So the diversity of outcomes you're getting with nucleases is much greater than with the base editor. And so we really like the purity and the predictability that you get with the base editor. In terms of your question about off-target overall profiling, you know, I think the field has actually come a long way. I think that we have great assays now to measure off-targets, both the nucleases and base editors. So I think we're getting, I mean, if you think about it, we have almost digital resolution here. There's only 3 billion things to check. You know, it's a gigabyte hard drive, and you just, you know, do the whole genome sequencing, you see if there's been any impact, then you can understand that with total resolution. And so for base editing, we've had to come up with a few extra assays that characterize single base changes compared to double-stranded breaks, and we've done that. And now we've been through the FDA with a complete IND, and they thought our package looked really good. So I think we're in very good shape there. And I would say I'm very confident and bullish about the sort of general off-target profile of the system.
John, I'm sure there are probably so many names behind the scenes that don't quite get the recognition they deserve. So Anzalone, Gadelli, Komor, some really superstar names. You know, they're superstar researchers, the headline being scientific achievements that most people are probably already aware of. Where do you give credit for being able to recruit top talent? And are there others that you'd like to mention today that, you know, deserve more recognition for pushing the boundaries of creative engineering at Beam?
Yeah, it's a great point. I mean, there's no question we've had some incredible science that has driven this, and science comes from people. I mean, these are just amazing minds. And so, yeah, so Alexis and Nicole, I think I mentioned before, were the pioneers who really invented base editing along with David Liu. Alexis is now a professor on the West Coast. Nicole actually followed the technology to Beam. And so she works at Beam as our head of gene editing platform technologies, and is an amazing leader. And in fact, I'd say of our first maybe 12 employees, probably 8 of them were from our founders' labs. And these are people who had seen the technology emerge and actually had worked on other technologies. So we have people who were at the early days or on the original papers of Cas9 itself, of CPF1, of many other sorts of innovations that came around at this time. So whether it be David's lab or Fung's lab or Keith's lab, we got some amazing talent in those early days. From there, you have to build a company, and we've really been growing, ever since. How do you attract the talent? I do think that the technology is so exciting. People who knew the field sort of knew this was a big deal and a potentially big breakthrough. I think the appreciation for working around double-stranded breaks, is taking more time to build up, but there were some people who said, yes, this is absolutely key. And then just the precision of the editing outcome. And then we just worked hard to create a culture where it was exciting for people to come and work. I mean, with some of these scientists, we're literally competing, not even with other companies, we're competing with that academic post where they can go be a professor or they can work at Beam. And so you think about how, you have to really create an exciting science environment to make that make sense career-wise. And I think we've done that. So I'd say early days, we had, Susan O'Connor joined us as our head of HR. She had built a lot of companies with Third Rock, and she knew how to do that and set up the right culture. So she and I worked closely together on that. And then Pino Charamella, our president and chief scientific officer, was also another early employee. And he really brought that knowledge of how you build this into a platform technology. And he came from Moderna. And in fact, at Moderna, he set up all of the vaccines division and is even on some of the patents of the stuff that became the bedrock for the COVID vaccine, which is pretty exciting. And so I often joke that Beam is sort of a mix of some of the stuff that I bring from Agios and then maybe some Third Rock in there, and then some Moderna from the platform. Hopefully pulling the best from all of those.
Really one Voltron.
Yes, exactly. That's the idea. But you can't predict how these things work out. I mean, a startup is a family, and it grows, and every single person who comes into the company adds something to that mix, and that's great. It just gets richer over time.
So you mentioned the idea of culture, and then also the idea of platform. So I really love that you brought those two up, and I plan to hit on those in a little bit. So as retail investors investing in early stage biotech companies, revenues don't always hold as much weight in comparison to established companies with approved drugs. So we have to objectively grade companies based on a variety of factors. And one of the most important factors is culture, as you mentioned. So although it's a relatively small sample size that you can see online, it's worthy of noting because it's clear you've built a one-of-a-kind culture at Beam Therapeutics, and it's uncommon to see 100% approval of a CEO, especially in comparison to gene editing peers in the space. So I want to briefly read off some of the employee reviews for you here. I think the audience will actually find this quite humorous when they hear some of the pros and cons noted by the employees, because in my eyes, the cons, most definitely could be seen as pros. So we're going to start off with the pros. Amazing research freedom, incredibly supportive culture, even for personal life and education goals. Great benefits and company parties and events, brilliant coworkers and collaborative atmosphere. The best science. Incredibly understanding of time off and work-life balance. Dedication to community and diversity, gives credit where credit is due in publications and patents, which that I totally agree with, because we see that time and time again. Management is excellent, with a CEO that actually has technical expertise, who makes it a point to know everyone and who contributes intellectually to science. Great culture, good benefit, good science, honest work, diversity is celebrated, never been talked down to or shamed. And so those are some of the pros that I've picked out and I wanted the audience to hear about. Now here are the cons. Sometimes conversations are too nice. Healthy and collaborative conflict builds better systems. Rapidly growing, so we'll have to see if they are able to continue the awesome startup culture. So John, can you tell us how important it is building not only best-in-class editors, but sustainable best-in-class culture where employees are able to show up every day and put in their best work?
Love it. I just feel like I got a review. That was great. I think it's such a great question, and it's so important, and we don't talk about it enough, frankly, as an industry. So there is nothing more important than culture. It's easy to say, but if you've ever been in a company that has a great one, or if you've been in a company that has a struggling culture, you know how important it is. I do think culture is very much of I think you pick it up from an apprentice model. I think, so it's just sort of seeing how it's done right. And so again, I think, I brought a lot from Agios. I think, others of our leadership team have brought things from their past, and we've just tried to put it together. One of the things that has typified the Beam culture from the beginning is it's very bottoms up. So our value statements, it wasn't me writing them down in the corner. It was actually, we worked together for 6 or 8 months. Then we said, okay, feels like there's something happening here that's good. Let's go off site and spend an entire day and a half and try to now articulate, what does it feel like to work at Beam? And that was actually the team that came up with these various sort of vision statement and values that we have. And I think that's been a way we've done almost everything. So we have so much grassroots, energy at the company. And we want to encourage that, right? I mean, because, we knew we were going to be growing and bigger. And so if it's going to be a kind of place where it's going to be top down, where I'm telling people what to do every day, not only is that not fun and people don't get excited to work there, it's actually not efficient. Like we knew from the beginning, the scale of what we were going to try to build was going to be really large. So it's got to be team driven and bottoms up, where people are taking accountability for the culture and for working together well and for living the values. So that's really critical. We do a lot of fun stuff too. I mean, so one of our values is fearless innovation. And so we try to really talk about risk taking and the fact that we're going to have to try things that may not have ever been done before. We may not have the answer on how we're even going to do it. And so, but we've got to try it, because that's where we're going to get these big breakthroughs. And so one exercise we did early days was we watched a lot of rocket launches. And so we watched all of the SpaceX videos where, they're trying to land the rocket and it's just blowing up constantly for 3 or 4 years. I mean, I can't even imagine how they stuck with it. And then finally they barely land one, and then they really landed, and then they land 2, and then they're launching a car on Mars, right? And so the lesson for me in that is that, 1, you have to be fearless, but 2, that once you're in these engineering loops, and I think gene editing is in an engineering loop where you're basically, hypothesize it, make it, test it, refine it, and iterate. These steady improvements, they become exponential in how they add up. And I really think that's happening with gene editing. I mean, look at Beam, right? We are an improvement on first gen technology that was only first published 9 years ago, and we're already reaching the clinic with a next gen version, and it's only going to continue, right? So we try to really capture that spirit at Beam, and above all else, we try to make it a real team environment where we're collaborative, we're transparent with each other, and where we're all aligned around this mission that we talk about, of creating lifelong cures for patients suffering from serious diseases. That, at the end of the day, is why we do this. And if we get stuck, the science isn't working, or if we're having conflict, or we don't know the path forward, coming back to that mission is going to be the thing that gets us back on track.
John, I love that you hit on that idea of like little iterations over time, incremental iterations over time, become, larger and larger changes that, ultimately, create more growth in a company. So the next topic I want to hit on is that you recently acquired the lipid nanoparticle discovery platform, Guide Therapeutics, for $120 million upfront and up to $320 million in milestone payments. So Guide was founded in 2018 and uses DNA barcodes to test hundreds of LNPs at a time for their ability to deliver genes to different cells and tissues. So during ASH, over the weekend, Beam, I believe, presented updated preclinical data using this technology to screen more than 1,000 LNPs for potential to deliver to HSCs, and identified LNP HSC1 as the most potent. So with efficient transfection in both mice and non-human primates. So there's a lot to unpack with this. So I'd like to approach it in several parts. Could you explain the DNA barcode strategy? What exactly is that? And how are you able to screen so many different lipid nanoparticles for delivery to different organ or tissue types?
Yeah, great. So we've talked a lot about editing. Editing is critical. We think base editing is a potential best-in-class technology, and everything about our strategy is designed to get it as broadly as we can, as fast as we can. So we famously are doing all of the different delivery modalities and therapeutic areas that are currently accessible in genetic medicine, we're going to go there because we think base editing has something to offer. So that leads us to delivery. And so in genetic medicine, delivery really matters. These are not pills. You can't just take a pill and have it go to the right cell. This is complicated machinery. You've got to get it in carefully. So we have a variety of ways to do that. One of our favorites is the lipid nanoparticle containing mRNA. And so what we put into it is that the mRNA encodes for our editor, so that once you're in the right cell, it'll express the editor, that's the protein. And then you also put in the guide RNA, that short little RNA element that's the targeting. And so those come together, goes to the nucleus, searches, finds, edits, and then washes out of the system and you're done. So that's a very complicated process that we need to trigger. So much like the COVID vaccines, we make lipid nanoparticles with mRNA in them, and that initially is going to go for our liver programs. And so if you put that into the bloodstream, it'll go to the liver, and we've optimized it so that we're getting very good liver penetration now, and we can indeed edit the liver with high efficiency, which is great, at least in primates so far. But LNPs are so good, we'd like to see them do more, right? You know, the great thing about LNPs is they're synthetic to manufacture, they're highly scalable, you can redose them, so you can, you know, add a little more, you're going to get a little more editing. They have a larger packaging capacity than some viruses. There's no pre-existing immunity that you have to worry about like some viruses, and ultimately lower cost of goods, which is, you know, if we're going to treat everyone around the world, which we would like to, that's going to be important over the long term. So we've been thinking about, could LNPs go to more places than just the liver? Now, the liver is the big, you know, sucking everything out of your blood. So that's an easy target. Everything else is going to be harder. But this company, you know, Guide and this team that had done some really pioneering work to create a system to try to screen LNPs to go to other places. And so basically what they did is we add DNA barcodes into that system that are unique. And you basically make a whole bunch of different recipes for your LNP, different cationic lipids, different formulations, different process, all of that matters actually. And so once you do that, you have tens or even hundreds of different lipid nanoparticles. You can then basically put all of them into the animal at the same time. And this is the really clever part. And so you're basically doing massive high throughput screening. I mean, that would be a single experiment every one you did otherwise. And then you basically look in your tissue of interest, right? So in our case, we might go to the blood stem cell and say, which of those LNPs got to this cell? And then you just read the barcodes out and it tells you, okay, these were the recipes that got them. And so, and then the even better part is you're screening the whole body every time. So it's not like I have to choose the blood cell. I can also check the muscle and I can check the brain, and we're going to learn that information, and you might get different answers, of course, right? Different LNPs may be good for different organs. So we are very excited about that. So the first lead formulation coming out of that work is going to be targeted at the blood. That's of course of great interest to us given our lead programs in sickle cell disease. So we're very excited about that. And so yeah, at ASH we showed we showed a little bit of mouse data early in the year. This time we showed expression of mRNA payload in up to 20% of the blood stem cells of a non-human primate at a clinical, relevant dose. And that is at or right at the front of the field, I think, in terms of people thinking about how to do this. And so we're very excited about that. That's obviously the subject of a lot more lead optimization to come. But ultimately, our long-term goal, our sickle cell programs will begin treating patients by editing the cells outside of the body and using a transplant to then put the cells back in. Long-term, we would love to switch that over to an in vivo edit where all you do is you put it into the bloodstream, goes to their marrow, edits, and they're done. And of course, there's many more things in hematology to do with that kind of technology beyond just sickle. And so it's obviously, very interesting. From there, the Guide platform, we are screening many other tissues, of course, that are of interest, whether it be T cells or CNS or muscle or heart, lung. I mean, these are all of interest, and we'll have to follow the science and see where we get the most traction and what that opens up therapeutically. It's not our only bet in delivery. We're actually doing a lot of different things in the delivery sector, but that's a big one, and we're quite excited about it.
So I'd like to hit on the idea of manufacturing solutions. So in November, you had a post on Twitter about the idea of scalability. You noted that at Beam, you strive to get these exciting technologies to more patients faster. You propose that one of the solutions is automation, and that you have several big robots running many experiments constantly and in parallel. You also posted a cool video of Beam team rovers in the automation lab. Could you share with us how you plan to leverage automation and robotics? And since then, has there been any update on the $83 million, 100,000 square foot biomanufacturing facility in Durham, North Carolina? And how might that spearhead partnerships down the line? I know that's a lot of questions. We can break it down.
Yeah, no, they're good. Yeah, so again, scalability is something we've been thinking about from day one, particularly, as Pino joined and we sort of talked about how are we going to build this thing. And the reason is because of the technology. We said, wow, we think that we have a big advantage here on the core technology, and we think there's a lot of ways we can use it. And so that means we need to scale. So, we talked about delivery then as sort of the next big component of that, which is where, how do we get as many organs as possible? The third leg of the stool is how do you make it? And so that's manufacturing. And again, genetic medicines, these are complicated things. There are not mature manufacturing contract organizations you can go to who've done this 1,000 times. There are some who do it, but they are in high demand, and it's still not your organization. Your science is developing in real time. And so we do believe that long-term, we need to internally control manufacturing and the know-how and capabilities and the team, frankly, the workforce to do it. Again, it all comes down to people who know how to do this well. And so we did make a decision that very early in our process, we would go and start to build out an internal capacity for manufacturing. That's in North Carolina. So as you said, 100,000 square feet, I believe that's the largest dedicated facility in the gene editing industry. We're building out in stages, but it's going to be quite significant. It can do clinical and ultimately also commercial. So that's slated to be online in 2023, and that's still on track. We do, of course, use external manufacturers because our current trials obviously aren't coming out of North Carolina yet, but over time, we'd like to see more and more go there. So that is a big part of our strategy for sure. And then automation you mentioned, which is interesting. Yeah, so that was another thing. We very early said, it would be really nice to have as much of this automated as we can, because then, our very brilliant and creative scientists can spend more time being brilliant and creative and less time pipetting, right? And that was a sort of initial idea, but it's actually pretty amazing how far it's gone. And so we basically now have an incredible automation team with these big robots. They're basically liquid handling, moving trays around from sort of different, machines, assays, process steps, things like that. We basically can do things like, let's say you edited a mouse, you can take a slice of the mouse liver, drop it on the robot, and then the scientist will get the genetic sequence from the sequencing in their email inbox. Like all of the processing and the isolation and the sequencing that happens is all automated, right? So that's incredible. So we're doing as much of that as we can. The rovers are cool. So they're basically they're our newest toy. They can give you more flexibility in how you move trays around. And then ultimately, as we get multiple big robots, they can actually drive from one robot to another. And then you kind of linking systems together. So my son is part of a Lego robotics team. And so we were taking a tour to sort of see that in action. And it's really pretty exciting. So automation is a big part of what we do. And again, all to try to do as much as possible, get more programs moving forward, and then ultimately, treat more patients.
So will those rovers be available by Christmas?
I think we're working with one of our vendor partners on this. I think we were the first company to actually implement those. So, wow. I think the whole culture of innovation at Beam is it suffuses everything. And we want to be as innovative in the automation and in the organization and the business as we are in the science. The whole thing should be creative.
So John, I just want to clarify, you mentioned commercializing your manufacturing. Does that mean that you maybe foresee other companies coming to you for their manufacturing, LNPs in particular? Is that what you meant by saying that?
Yeah, it's a good question. There's probably different ways we could talk about that. So one would be just our own medicines eventually, right? So if we ever get an approval, we will decide which medicines does it make sense for Beam to commercialize, and then we would be able to manufacture that for commercial supply out of that facility. So that's an important factor. But actually, and you would ask the question about partnering, which I didn't answer, but yes, a lot of the part about scaling and having this high throughput is it means we can do a lot. It will be more that we could do than we can prosecute ourselves in development. And that's going to be true. But what that does, it creates opportunity for partnering. And now we can put programs in other people's hands that they might even be better at moving forward than we will, or that we together can do something even more special. And then again, increase the total amount that can move forward and the impact we can have. And so yes, at some point, maybe that does include the manufacturing side where, you can come to Beam and you can get it all. You can get the editing you can get the delivery, and even we can make it with high quality. And I think that's important. I think that there's still a lot of fragmentation in this field. And for me, if there's a compelling bit of biology or a patient you want to treat, it seems crazy that you have to do 3 different arm's length deals just to put together one program concept and then start to move it forward. It's much better in my mind to have all of it under one roof, and then we can put it all together, and then you're off to the races, moving to the clinic.
Sometimes we joke on Twitter about like Beam is going to be base editors as a service, sort of like we think of software as a service. Maybe it will be LNPs as a service. Who knows?
I think that's not crazy. I mean, I think we have been. I think we do a lot more in our platform in terms of creative business development and trying to sort of do clever ways to make more things happen than certainly any other company that I've seen recently. And I think we only want to do more of that, especially as we continue to put all of this technology and capability into one place. We'd be crazy not to have that be a big part of what Beam does.
For sure. I'd like to discuss 2 of your lead hematology programs, Beam 101, activation of fetal hemoglobin, and Beam 102, the Macasar variant direct correction of sickle cell causing mutation. Could you describe the benefits of both strategies, some of the differences between them? And do you plan to move forward with just one or hopefully both in the treatment of sickle cell disease and beta thal? So could you talk us through your decision process there?
Yep. Yeah, so we have 2 programs for sickle cell disease. Beam 101 takes the now clinically validated approach of raising fetal hemoglobin, which is a kind of hemoglobin that turns off when you're 6 months old, but if it's on, it can compensate for the presence of the sickle protein, the sickle globin that's causing the disease in sickle cell. And so we have one that is without cutting and non-viral. It can go in and it literally changes single letters in the on-off switches of the fetal hemoglobin genes, turns them back on, and we get very high levels of editing over 90% and very high levels of F over 60%. And then concomitantly, the S level, the sickle protein, is turning down. And so we're down around 40% or less sickle protein. And that's actually the same ratio that you get with a patient who has sickle trait, where they have 1 copy of sickle and 1 normal copy of hemoglobin. So we think that's quite important, and it's actually a slightly better profile than what others are getting in the clinic. And so we think that's a potentially best in class way to do that fetal hemoglobin strategy. Beam 102 is the direct correction. So we go right to the adult hemoglobin gene that's mutated. Every patient has a single letter misspelling, and we can edit it back to a normal copy. And so that's super exciting as well. And so now you're literally creating cells that don't have the sickle protein at all. And so when you look at the total edited cells, where editors, we're editing over 80% efficiency, you're down to about 10% S in total, and everything else is normal hemoglobin. So that's very exciting. So we love both of our children. I think they're both potentially best in class products. We'd be thrilled to have either one of them, and we have both. So our basic plan is to not try to overthink it and just follow the science. And so we're going to move both of them into the clinic. We'll get data with both, and then compare and contrast, and we'll figure out. I think it is likely that at some point in development we will make a choice of which of these 2 will we register for approval in sickle. In the case of 101, that can also treat beta thalassemia because you're upregulating that fetal hemoglobin. So that's another possible path that program may take. And so that's all part of our clinical development program. And then, as I mentioned before, in parallel to those paths, which we're quite excited about, we will also be working on conditioning, which we want to basically make that regimen for the transplant less toxic. And we have a lot of ideas for that, which we think can make it better. And then ultimately start to think about in vivo delivery, as we talked about, where you're getting rid of the transplant altogether. And so this is a really long journey. But the nice thing is those same Beam 101 and 102 editing payloads would be the same ones that move into those waves 2 and 3, where we're thinking about better transplant or in vivo delivery. So this is all really one integrated strategy to try to create what we think is the best regimen to cure sickle cell patients, here and abroad.
And John, I just want to add, because I think we skipped over just a little bit. Is it the Macasar? Am I pronouncing it correctly?
Yep.
Macasar variant is not actually the wild type variant or the wild type gene, that most of the population has. With your deaminase, you're actually able to change it to the Macasar variant, which is also a functional copy. Could you explain a little bit about what you're doing? Is it still expected to be just fine in patients?
Yes. So basically, the edit we make with the A base editor is an A to G edit. There's a class of mutations called transition mutations, which are A to G and G to A, and then C to T and C to T. And those are the edits we can make with the base editors back and forth, based on the chemistry that we're doing literally on the genome. So that represents about 30% of all point mutations, or 30% of all total mutations, and well over half of all point mutations. So there's a lot to do with those. The sickle mutation is not in that group, actually. The sickle mutation is an A to T or a T to A change, okay?
Yeah.
And so we can't do an A to T with the current A base editor. We would need some different chemistry to do that. But it fortunately turns out that the A to G change we can do, it gives you a different allele, but one that is normally occurring in the human population. It is not the dominant one that most people in the audience will have, which would be a T. But with the G, you get this thing called HBG or Macasar. And basically the punchline, as you said, is it is a normal functioning hemoglobin. So it's about 0.1% of the human population probably has it. And actually at ASH, this is the other data update we gave, just to be, I mean, for 40 years, people have known about this form of hemoglobin and that it is normal, but we want to be made really, really sure. And so we've been doing all sorts of different studies to sort of really show that it is indeed superimposable on the normal, more prevalent copy of hemoglobin, and indeed that is the case. So we fully expect this to be a curative conversion of the mutant protein to the normal.
And so, John, you also mentioned that idea of conditioning. So can you explain the importance of your non-exclusive partnership with Magenta for their targeted condition approach to reduce toxicity challenges associated with the standard of care? How is it differentiated from traditional chemotherapy or radiation regimens? And why do you feel this partnership was in your best interest?
Yeah, so we like the Magenta program a lot. So the basic idea here is you need to make room in the body for the new edited cells to come in. And so you can do that with chemo, which kind of wipes out all of the cells that are there, and there'll be plenty of room. But in the process, it's doing a lot of genotoxic damage, and it's just harsh on the patient. So what we'd like to do is have a less toxic regimen. And so the idea here is to use, much like precision oncology therapies, to use antibodies that can go right in and target, in this case, the hematopoietic stem cell itself, and try to get rid of that, but not have such a deep impact on the other cells around it, and to get rid of it, and not using just a chemical warhead, but something that's a little more sort of smart. And so Magenta has a really nice program targeting the KIT receptor on HSCs that looks really good. That's moving into the clinic now. They're going to begin in AML, but their long-term vision is to bring that into transplant. So we think that looks really, really interesting. But one way or the other, we want to make sure we get to this sort of vision that there will be a less toxic conditioning agent for transplant, because what that means is it means that more patients will opt for the therapy, right? So in Gen 1, if you're using chemo, there will be some number of patients who have severe disease who are going to definitely want to get these treatments. And I can tell you the demand for clinical trials right now is very high. But there will be another set of patients who look at the transplant and say, that doesn't make sense, and this just doesn't feel like the right time for me. And for them, I'm quite hopeful that we'll have good small molecules and antibodies and things like that, and those are coming, which is great. If we can make conditioning better, we will shift the balance where more and more people will start to opt for a curative genetic therapy. And then, of course, long-term, that in vivo editing, I think, will even more. It's just that much more patient-friendly and easy to administer. So this is, again, back to that long-term strategy to create the best regimen possible.
So for Magenta, those antibodies, are those used as sort of like delivery vehicles to for that targeted conditioning? Is that how the antibodies are used?
The antibody, it is the conditioning agent, actually. It basically it goes in your bloodstream, it'll find the HSCs, bind to it, and then go in, and then it's got a payload that will get rid of the HSC, but in a very targeted way because of the targeting of that antibody.
Got it. So Beam 201 for the treatment of relapsed refractory T-cell acute lymphoblastic leukemia is a potent and specific allogeneic CAR T targeting CD7 malignancies, which uses multiplex-based editing to simultaneously make 4 knockout edits at over 96% efficiency for each gene. Relapsed refractory T-ALL is a severe disease affecting children and adults with a 5-year overall survival rate of less than 25%. What excites you about your differentiated platform for T-cell editing, and what do you say to opponents who believe that you might be at a disadvantage in terms of using knockouts and not using a double-strand break to insert CAR into the track locus.
Yep. Yeah, so this is now our second big therapeutic area after sickle. So immunology and thinking about cell therapy, making things like CAR Ts. And so, here we're taking advantage of the fact that base editors don't create the double-stranded breaks. And what that means is we can stack edits on top of each other with virtually no limit and have no consequences.
Wow.
Whereas if you do that with nucleases, you're making a cut, you make another cut, you make another cut. Now the cell is even more challenged knowing how to put the pieces back together again. And it's going to be quite alarmed to see all of this genotoxic activity happening at the same time. And so what happens when you get to 2, 3, especially, and anything beyond 3 number of edits is if you're using a nuclease, you get lots of DNA damage pathways that turn on, you get cell viability loss, and you get chromosomal translocations and rearrangements. And that's where these pieces are getting put together in the wrong order. And that's quite concerning. So with base editing, we get none of that. I like to joke that it's almost as if the cell doesn't know it has been edited. Because it's a very subtle process. And I think, from a pathway, gene regulation perspective, it looks like that's true. And so with Beam 201, we want to take advantage of that. So we made a cell product that has 4 different edits. This will be the first quad edited cell product in the industry, I believe. And for a target, we chose T cell leukemia. And so B cell leukemia, acute leukemia, is the place where all the CAR Ts are working, CD19 famously. But the T cell side was left alone. One of the reasons it was left alone was that in addition to edits to track and other things, you had to also get rid of the target of your CAR, because otherwise your CAR T cells will attack each other before they have a chance to attack the tumor, and that's called fratricide. And so it just needs a lot of editing to get this to work. And so that's right up our alley. So we think there's a great demonstration for that. So one of the edits is to knock out CD7, so that when we put in the CD7 CAR, the cells will be perfectly happy until they get in near the tumor and then they'll attack, right? And so I think it's a great program. It's very exciting. We're knocking out 4 different things. And these are patients who really don't have good options. And so they desperately are in need of new therapies. Beyond this, there is just an infinite landscape of possibilities in cell therapy. I think cell therapy and editing are going to go together forever. And I believe that as the cell therapy landscape advances, people are going to want to basically add more and more edits, because you're going to want to add more functionality. The more we learn, the more we want to do to these cells to make them better. That's going to play to base editing's advantage, where you're going to want to use this multiplex. Now, the last piece is the CAR, which you mentioned. So in terms of getting the CAR in, there's really 2 options. One is lentivirus, which is the standard that Bluebird used, and Juno and Kite and all those companies. And then editing, you have the opportunity with the double-stranded break to create a cut, bring in a template, and then insert it at that cut site. And specifically, people have liked the idea of putting it in the track locus, so that instead of expressing track, you're expressing the CAR, and maybe the regulation will help a bit. So that's one sort of special component. So we will always do multiplex editing with base editors to knock out as many things as we need to. And then for the CAR knock-in, we have both available to us. So we have a nuclease called Cas12b that works very well. And in fact, we just partnered it with Sana to do some really cool cell engineering work, and they do need to knock things in. And so that made sense for them, and it works. For Beam 201, Lenti is already well validated, and we felt that was an easier path forward. And we are not yet convinced that there's a big benefit from the track insertion. I think we'll see. We're just sort of watching the data accumulate. But if it turns out that track insertion is going to give you an efficacy boost of some kind, then we can shift the platform over there. But I think Lenti works great as well, and is the standard of care for a lot of CAR T products. And so that's where we were for 201.
John, what have you seen that makes you feel so confident that you can stack more than 4 edits or 5 edits, and maybe even go up to 8, 9 edits? What makes you feel confident that you guys can actually do that? Have you seen anything preclinically?
Yeah, well, we've done 8 actually.
Okay.
And it still works. Wow. I don't think we've bothered to go much past 8. I think the problem, you get to that point, I think actually our editing capabilities are beyond our biology insight. Okay. Right. I mean, cancer, I like that. Cancer and immunology are complex systems. So there's still got to be the careful work of saying, what do we want to edit and why, and how does it work? But right. I think we published quite recently a kind of next gen program idea where we're looking at CD5 for another form of cancer, T-cell lymphomas. And there we use 5 edits in that product, and we showed that data. So yeah, it can keep going for sure.
So part of the investment thesis for Beam is that not only is your company bringing potential best-in-class editors and therapies to the pipeline, but that you also have a platform where you can out-license your technology and benefit from downstream royalties and equity. So as you just mentioned, a couple minutes ago, could you talk to us about your partnerships with Verve, Apellis, and Sana, like you just mentioned? Is this out-licensing strategy a big part of what Beam is planning to do in the future?
Yes, it is. I mean, so the steps in the strategy were basically, okay, we have this incredible technology. We're really excited. We want to bring it broadly. These are complex systems where you need payload, delivery, manufacturing, and we don't want to have to cobble that together every time. So let's get all of that under one roof. Let's go deep into science and innovate to make even these things better. So we're doing more editing work. We have a nuclease. We have other things as well. Delivery, we're innovating there. Manufacturing, great. Once you've done all of that, as I said, you have way too much in the platform to do yourself. And so we've got to do partnerships that gives us the chance to sort of say, here, these are the things that would fit best within Beam, and here are some opportunities to put it outside. So the 3 you mentioned are great examples. Verve was the first one. And in that case, they were already a company, basically a group of really amazing cardiology experts had this vision to use gene editing to do a permanent knockdown of cholesterol pathway and effectively eliminate or drastically reduce your later risk of any heart attack. And it's an amazing vision. And it shows you, I think, how, although we're beginning in rare genetic diseases with severe mutation status, this technology, as it matures, is going to move relatively quickly to more common diseases and to even things like disease prevention, risk modification, et cetera. So I think it's that's one of the things I see happening, in the coming decade. So with Verve, we gave them all of our technology. They ended up doing a bake-off between nucleases and base editing, and chose base editing because it was more potent, more precise, and worked great. So their program is now moving forward, targeting PCSK9. And then Beam has an opt-in right in the US for a 50-50 split on that product, which is really exciting. And so it's a good example where we can, we don't get money from them, but we get some strategic value and rights later that we could participate in. And a great product moves forward that we probably wouldn't have been equipped to do because we don't have that cardiology expertise. Apellis, very deep in complement biology, complicated pathway. But they had really interesting ideas about how to think about editing, and we wanted to work with someone like them on what does it look like to intervene in a complicated biological pathway using editing. And so that's a great program and a great deal with them. That obviously brought in money, so we have a $75 million deal there, and again we have an opt-in to share of the US rights on one program. And then Sana is working on allogeneic cells and in vivo delivery, but for their ex vivo programs, they need to do a lot of editing. And so we did a deal with Cas12b, which is a nuclease that we weren't doing too much with, other than that idea of maybe use it for a CAR T insert. And so, but Sana can do a lot with that in the near term. And so that was a great deal, 50 million upfront, non-exclusive. So Beam is still able to use Cas12b for any purpose, but it gets Cas12b moving forward and start to get developed. And so that's pretty exciting too. So yes, I think you should definitely expect that Beam will do a lot more of these kinds of things. We try to be very creative on how we approach these situations. It's not a cookie cutter approach, but anytime that I think there's synergy with someone outside who has a great team or great capability and anything we have that can help, unless we're doing it ourselves, that's something that we would like to see happen. And I think this will be a real distinguishing feature of the Beam strategy over the coming years.
I think it's just more evidence that we love to call the ultimate deal maker. We constantly see Beam coming out with these amazing partnerships with companies that we all love. Verve, Apellis, Sana. These are companies that we follow and companies that are doing really, really great things. I did want to ask you, John, is Verve going to continue to be using the base editors, or is it just with the PCSK9? You have any insight on that?
Yeah, so they actually are also using base editing for their second program, ANGPTL3, which is also very exciting, and we have that same opt-in to that program as well. There are another couple targets where they have rights to our stuff as well. So I think they certainly are in the next program at least, and they may well in the future. Verve has always said they're technology agnostic, and so they're just looking for the best technology for a given application. So I mean, I leave it to them to sort of go target by target and decide what the best editing strategy will be. But certainly for the first couple, it looks clear they're going to be using the base editors, and I think that those programs look really strong.
Got it. Awesome. Thank you for that. And so your lesser known RNA editing platform, Repair and Rescue Systems, where Repair is capable of A to I editing and Rescue is capable of T to U editing. Where do you see this platform really shining in comparison to the DNA editing that you can do?
Yeah, so some of that editing innovation I mentioned beyond base editing, we got Cas12b from Feng Zhang's lab, and then we also got these RNA editing systems as well. And so they're very interesting. They basically use a different CRISPR that can target RNA strands instead of DNA strands. And then once there, use a deaminase to do a sort of single base edit, which is very interesting. The whole field of RNA editing, I think, is still earlier, obviously, than DNA editing. And it has a different feature. Basically, the edit you make is now transient. So it's not going to last for the lifetime of the patient. It's going to last for the lifetime of the mRNA that you've edited, which will be a few days, right? And so that's the key difference. And so you have to think of basically one of two paradigms here. And we're not the only ones thinking about this. There are other companies in this field. One would be, do I express the editor forever? Or do I chronically dose it so that I'm constantly editing the mRNAs as they come out to make some kind of an impact? And that can work, although you now have the challenge of it's a chronic therapy and you're basically delivering this forever. The other thing you think about is transient delivery to make a transient impact. And so something like regenerative medicine, which we've talked about before, where maybe you want to change a cell's behavior for a while, drive it in some new direction, but then you want to let go and have it stop. So that would be a really interesting application for RNA editing as well. And so we're doing some of these things in our platform on this technology, but I think it is definitely much earlier stage. I think for a lot of the diseases that people know us for, whether it be sickle or the liver diseases, things like that, I think all things being equal, I think if you can do it with a DNA edit where it's a permanent change and you've given a lifelong cure to a patient, I think that would be preferred. And so that's kind of our primary focus, I think. But RNA editing, I think, will have a role to play, and we're exploring that.
And so last but not least, and perhaps one of the most exciting things about Beam is your partnership with Prime Medicine, which is currently still mostly in stealth mode. You and Dr. Liu have mentioned, a divide and conquer approach. Could you talk us through that close relationship with these two companies and, how you plan to pursue Prime Editing in your own portfolio? And do you foresee that these will remain separate entities for the foreseeable future, or any hopes of profit sharing by any chance?
Right. Yes, I think they'll remain separate entities for the foreseeable future. So yeah, Prime is very exciting technology. It falls into that sort of same next gen category that base editing falls into, where the similarity is we're going to get rid of the double-stranded break, we're going to target the DNA, and we're going to bring along an effector that's going to do something once you're there that's very precise. In the case of base editing, it's a deaminase, where we make a chemistry-driven single base change with high efficiency. In the case of Prime editing, you're bringing along a reverse transcriptase, where you're going to try to rewrite a short section of DNA and have that get back into the gene and sort of do local rewriting. I think there will be other categories. There are people using transposases of various kinds to try to insert large genetic elements into genes. So I think that this general feature of we've separated out the targeting from the editing elements, and now you can independently mix and match and optimize those is going to be a big feature for the future. And then I think you're going to have sort of a wide toolkit of different types of tools. If you want to make a very precise single base change with high efficiency in any cell type, base editing is going to be it. If you want to make more flexible local rewriting, Prime is going to be what you want to use. If you need to drop in an entire gene, it's probably something like a transposase or other sort of technology. So we see all of these sort of having a role to play in the future. So with Prime, as it came up, we worked with David and actually with the same investor set that we had at Beam to do something that I think is pretty innovative. Back to the idea of trying to be as innovative on the business side as we are on the science side. And so we said, well, look, this is really new technology. It's going to need its own build. But at the same time, it has some application that could be similar to what base editing is doing. And we have a lot of Beam that might be helpful to exploit for the technology. And so what should we do? And so what we did is we set up a new company. It's called Prime Medicine. We were all of the interim leadership of that company in the early days, and I'm still on the board. David obviously was the founder. And then the same investors invested in it. And then that company took down the editing rights from the Broad. And then we did a deal between the two companies. And so the deal basically says that Beam will share delivery technologies and other things with Prime and help set it up. And then we take, in return for that, exclusive rights to use Prime editing technology for any edit that is similar to what base editors can do. And that's that 30% of the correctable mutations that I mentioned before. And so that's a lot of territory, which is exciting. And then Prime takes the rest of it. And so the idea is that divide and conquer idea is that basically it sort of means that we can double down on our target space using Prime if we need to, and then Prime Medicine will focus on different diseases than we are. And I think when people see the Prime Medicine pipeline, you'll see that is in fact exactly what's happened, which is I think a good sign that this sort of creative structure is working. I think, still early days with Prime technology, but a lot of progress being made, and we're certainly excited about that for the future, along with, I think, a lot of other interesting systems that are coming online. I think if I could draw an analogy, I think that it's sort of like we're in the early 80s with computers, right? And we've now got the first laptop, or we've got the first personal computer, and they're amazing, but they're also going to keep changing, and these things are going to keep evolving over time. And not only that, but when we think about where we will be in 10 years or 20 years, it's going to be mind-blowing compared to even where we are today. And so the most important thing for us as a company and others is to be part of all of this, and to stay relevant and stay on the cutting edge of these different technologies, or to build the delivery engine, right, that through which any genetic payload can be delivered, right? So that's that sort of critical mass we're building in all of the integrated capabilities to do what we call precision genetic medicine well. That's really the strategy. And so, obviously base editing is a big part of the future. And I think it is working so well. I think it'll be probably the majority of what we do for the medium to long term, but very long term, ultimately we're flexible, and we want to build really a team and a capability and a company that's capable of leading in this field for the long term.
So will Prime be staying away from your specific indications? And do you see that maybe, once the time comes that you can create future iterations of this therapy or therapeutic with Prime? Is that kind of what you're saying, for future iterations of sickle, for example?
Yeah, so Prime will stay away from our indications. So only Beam has the rights to use Prime and sickle. And then the other diseases in our sort of universe as well. I think we've got it if we need it. I think that, again, base editing is working really well. So I think I wouldn't expect that Prime displaces base editing in a lot of these areas. I think it maybe comes in handy, if we have maybe a challenging base editing target, Prime can solve that given its flexibility. That would be great, right? So that's where it expands our toolkit. Which is quite exciting. But I think it's, I think the companies will basically have, different applications from each other over time as we apply this in different areas.
Gotcha. Thanks for clarifying that. And John, the last question. A lot of people are always asking me, when are we going to be able to be buying merchandise from Beam? When can we buy sweaters and hats, sweatpants and things like that? Will we ever have like a merchandise store where, your fanatics can purchase apparel?
All right, that is a great point. I'm going to think about that, and I will get back to you. And if we do it, you'll be the first to get the email.
Awesome.
For sure.
Awesome. We can't wait. Okay, everybody. Thank you so much for tuning in. I hope you enjoyed that.
Thanks, Nat.